Rub your thumb across a fresh blueberry. The dusty blue comes away, and the skin underneath is almost black. The blue was sitting on the surface the whole time. It was never soaked through the fruit. Now set a blue jay feather and a hydrangea petal down next to that berry. All three read as blue, but no two of them are blue for the same reason, and one of them is barely carrying any blue at all. So what is each of these things actually doing to the light? We usually treat color as paint. A strawberry is red, so we imagine red stuff in the strawberry. A leaf is green, so we imagine green stuff in the leaf. That picture works often enough that it starts to feel universal. If something looks blue, surely it is blue for the same simple reason. But blue is awkward. Biology has many good ways to make dark colors. It has broad roots into browns, blacks, reds, and yellows. Those colors can come from pigment families that absorb a lot of incoming light and leave the rest for our eyes. That is a fairly forgiving job. A surface can be messy and still look dark, and a pigment can be broad and still look rich. Vivid blue asks for something fussier. A blue surface has to send blue light back at you while not sending back too much of its neighbors. That is a narrower optical result. It is less like throwing a blanket over the light and more like keeping one clean note while muting the choir around it. You do not need a full spectrum lecture to feel the difference. Black can happen when lots of light gets swallowed. A sharp, bright blue needs more selectivity. So the first big correction is simple. Blue is not just another color job. That does not mean nature never makes blue chemistry. It does, and flowers prove that later. But a cheap, durable, all-purpose blue pigment is not something life seems to reach for as easily as other color roots. And once that root gets hard, evolution starts changing the question. Instead of asking, how do I store blue paint, nature often asks, how do I build something that looks blue? That is the engine for almost everything that follows. One root is structure. Arrange matter on a tiny scale so light gets scattered or reflected in a very selective way. Another root is surface design. Put a thin outer layer where the visible color lives at the surface instead of deep in the object. And another root is tuned chemistry. Use pigment, yes, but then surround it with exactly the right conditions so the visible result slides toward blue and stays there. So when you meet a blue thing in nature, the useful question is no longer what blue dye is inside it. It is what this object is doing to light, and the cleanest place to see that trick in action is a feather. A blue feather looks dyed, and that is what makes the reveal so satisfying. On the surface, it feels obvious. There is the feather, there is the color. End of mystery, but many bluebird feathers are not carrying a simple blue pigment the way a red petal might carry a red one. Inside the feather barb, the color is being built by a nanostructure made from ordinary material, arranged in a very particular way. Keratin and tiny air pockets create a microscopic architecture that handles incoming light selectively. Blue survives the trip to your eye. A lot of neighboring wavelengths do not. That is structural color. Once you see it, the feather stops looking painted and starts looking engineered. It also helps explain why the blue sits where it does. You are not looking at a pot of blue liquid trapped in a barb. You are looking at a surface whose tiny physical arrangement makes blue the visible outcome. The color is less like ink soaked into paper and more like a device quietly doing one optical job very well. Butterfly wings push the same idea even harder. A blue morpho can look almost offensively blue. It flashes so brightly that the obvious explanation feels unavoidable. Surely that wing is loaded with some brilliant pigment, but tilt the story open, and the trick gets much stranger. Underneath that famous blue, much of the wing material is brown. The electric blue is the visible product of microscopic ridges and layers that manage light at the surface. That is a brutal little payoff. Something can look as if it were painted with the purest blue in the world, then turn out to be brown plus architecture. And that matters because it stops the whole idea from becoming a bird curiosity or a butterfly curiosity. It becomes a deeper pattern. Evolution does not need one magic blue substance if it can keep building surfaces that return blue selectively. Spiders make that point beautifully. Different blue tarantulas can land in similar visible blues even when the nanostructures involved are not copies of one another. So the repeat is not one exact design. The repeat is the strategy. Different instruments, same note. Across feathers, wings, and spider hair, the common move is not to store blue. It is to build a tiny structure that makes blue happen. And once that idea is in place, the next step is even better, because some animals do not just hold that structure still, they tune it while alive. A chameleon is often used as a generic color change mascot, which is a shame because the mechanism is much more interesting than the mascot. In panther chameleons, part of the color shift comes from superficial cells containing guanine nanocrystals. Change the spacing between those crystals, and you change which wavelengths are most strongly reflected. So the color change is not only about pumping in a different pigment, it is also about rearranging a microscopic optical system. That is a wild upgrade of the same logic. The feather gives you a built structure. The chameleon gives you an adjustable one. Blue becomes something a living tissue can dial by changing crystal spacing. The visible color is still a surface-level answer to a hard optical problem. It is just being tuned in real time, and that keeps the story honest. Structural blue is not one museum trick that belongs to a handful of dead specimens. It is an active biological strategy. Mammals make the point from the opposite direction. If simple blue pigment were cheap and easy, you would expect mammalian blue to be ordinary. Fur, skin, and tissue would use it everywhere. But vivid mammalian blue is rare and specialized. In cases like mandrill facial skin, the visible blue is again tied to tissue structure and light scattering rather than a flat patch of blue dye doing all the work. That rarity is evidence. It tells you blue is not a default setting waiting to be turned on. Mammals are full of color cues, yet straightforward blue still tends to arrive as a special construction project. This is where a topic like this can go wrong if it loses discipline. The easy failure is to collect animals the way a scrapbook collects stickers. Bluebird, blue butterfly, blue spider, blue lizard, blue monkey. Cute, varied, and completely forgettable. But the species are not the real repeat. The mechanism pressure is. Across very different bodies, blue keeps showing up when matter is arranged so light is filtered, scattered, or reflected narrowly. The exact physics is not identical in every case, and it would be sloppy to pretend otherwise. Still, the family resemblance is strong enough to matter. Nature keeps returning to built surfaces because blue keeps refusing to behave like easy paint. That is why the middle of this story should feel tighter, not broader. We are not wandering through a zoo, we are watching the same problem get solved again and again with tiny construction. And then the plants arrive to stop us from getting too comfortable because flowers can make blue too. They just do not make it the easy way. Blue flowers look like a counter-argument. You hold up a hydrangea or a delphinium and the whole structural color story seems to wobble. There it is, a petal, a soft tissue, a blue color. Surely this is the moment where biology simply makes blue pigment and gets on with its day. Not quite. Plants do have a chemical route toward blue, often through anthocyanin chemistry and especially delphinidin family molecules. The point is not to deny pigment, it is to show that even when plants reach blue by chemistry, the job is still unusually delicate. Hydrangea is the perfect reveal. Its blue is not just one molecule acting alone like paint squeezed from a tube. The visible color depends on the pigment plus the chemical environment around it. pH matters, metal ions matter, companion molecules matter. The petal does more than hold blue. It holds a small chemical arrangement in a state that pushes the visible result toward blue and keeps it there. That turns a flower into a systems problem. The difference matters because it preserves the same deeper rule we saw in animals. Blue is still fussy. In a feather, the fussiness is physical architecture. In hydrangea, the fussiness is tuned chemistry. Either way, the color is not arriving cheaply, and once you know that, blue chrysanthemum stops sounding like a gardening side quest and starts sounding like evidence. Breeders and researchers have had a hard time pushing chrysanthemums into a stable blue for exactly the reason this whole episode exists. Blue is not a simple switch. It often needs multiple biochemical conditions to line up together before the color really lands. That is why flowers do not break the argument, they strengthen it. Yes, biology can reach blue through pigment chemistry, but even there the route tends to be narrow, conditional, and easier to disturb than a broad, common color family. The flower is not saying blue was easy all along. It is saying we found a chemical way to do it, and even that way needed a careful setup. So by now we have two strong branches. Animals often solve blue with structure. Flowers can solve blue with tuned chemistry. And in both cases, the visible result is less like ordinary paint than it first seemed. Which brings us back to the kitchen bench, because one of the most familiar blue things in daily life is hiding the trick right on its skin. A blueberry seems like the least suspicious object in the whole story. It is small, it is familiar, it is called a blueberry. You would be forgiven for assuming the blue goes all the way down, but the visible blue appearance can live in the fruit's outer wax bloom. That thin surface layer scatters light in a way that gives the fruit its dusty blue look. Disturb the bloom and the visible color changes with it. Which means the blue is not simply stored in the obvious way, like paint mixed through the flesh of the fruit. Once again, the color sits at a surface doing a light job. That is such a neat little ambush. The berry looks like the plainest pigment example on the table, then turns out to be another optical workaround living in a thin outer layer. And because it is a household object, the lesson suddenly stops belonging to museum specimens. You can imagine rolling the berry between your fingers and watching the mystery move with the surface. Blue eyes do the same job from even closer range. If someone has blue eyes, the obvious story is that the iris contains blue pigment. But human blue irises are not blue because they are painted with a blue dye. Here again, light handling matters more. With relatively low dark pigment in the front layers, scattering helps blue light dominate what you see. So even a blue eye is another answer to the same question. Not blue paint, blue outcome. That does not mean a blueberry, a blue iris, a feather, and a hydrangea all use one identical mechanism. They do not, and that is exactly the boundary worth keeping. Surface bloom is not feather nanostructure, iris scattering is not hydrangea chemistry. But the naive assumption fails in every case. The finished blue keeps hiding a more complicated backstage job, and that is where the opening objects pay off. At first they sat on the table as one color family. Now they split by mechanism. The feather is built, the hydrangea is tuned, the blueberry is surface blue. The eye is scattering blue. The label stayed the same. The explanation changed underneath it. Once you notice that split, blue becomes strangely rereadable. You stop asking which animal or plant you are looking at first. You start asking what kind of solution you are looking at, and that is enough to say the bigger thing cleanly. These are not disconnected exceptions. They are signs that evolution keeps running into the same problem. Now the pattern is clear enough to say in one line, blue keeps appearing in nature, but the route to blue keeps exposing hidden engineering. Sometimes that engineering is nanostructure in a feather, wing, or spider hair. Sometimes it is a tunable crystal array in skin. Sometimes it is a wax bloom on the outside of a fruit. Sometimes it is an iris where scattering does the work. Sometimes it is flower chemistry balanced so carefully that the color depends on the whole setting around the pigment, not only on the pigment itself. Different route, same pressure. Straightforward, durable, all-purpose blue is not an easy biological color job, so life keeps building around the difficulty instead of making it disappear. And the rarity itself becomes evidence. If vivid blue were as chemically easy as dark brown or black, you would expect it to spread through the living world with the same casual abundance. Instead, it often arrives in sharper, more specialized forms, flashing from wings, sitting on a surface bloom, appearing in petals that needed a carefully tuned chemical setting, or showing up in tissues that scatter light just so. That patchiness is telling you something. It tells you blue is often a precision job. There is a temptation here to flatten the story into a slogan. Nature fakes blue. That is catchy, but it is too blunt on its own. Some flowers really do reach blue through chemistry. The better takeaway is subtler and more useful. Nature often cannot rely on one simple blue paint, so it keeps constructing blue through structure, surface design, or tuned chemistry. That version keeps the exceptions honest and the pattern intact. It also changes how ordinary beauty reads. A blue butterfly stops looking like someone found the right pigment and splashed it on. A blueberry stops looking like blue flesh wrapped in blue skin. A hydrangea stops looking like a plant that casually happened upon sky-colored paint. Each object becomes a record of work done behind the scenes. And that is why blue feels different from many other colors. The finished color looks calm. The mechanism underneath it usually is not. It is selective, layered, and tuned, built on very small structures doing very exact things to light. Once you start seeing that, the question follows you out of the room. Not, is this beautiful? That part was obvious already. The better question is, what had to happen for this blue to exist at all? At the start, a feather, a berry, and a petal all looked as if they belonged to one easy category, blue things. Now they do not. The feather was building blue with structure. The berry was wearing blue at the surface. The flower was tuning chemistry into a narrow result. And even a blue iris turns out to be another light-handling workaround instead of a neat layer of blue dye. That is the real twist. The strange thing about blue in nature is not that it is missing, it is that it so often has to be assembled. Our eyes only see the finished color. Nature was dealing with the harder job underneath it, solving a chemistry problem with tiny architecture, delicate molecular tuning, and thin outer layers that quietly do the optical work. Blue stays beautiful, it just stops looking simple.